This is the Soil Health Indicator webinar series, and we're on webinar 3 today, so welcome. The webinar from Wednesday on soil health and soil health sampling and kind of strategy for testing is posted online, and you can see on the slide being shown where you can find those recordings. And Joshua Faulkner presented yesterday on physical properties and the indicators of physical properties for soil health. And I believe his webinar is posted as well. So those can be found online if you missed them, and they'll be there for future reference too. Again, if you are looking for CCA CEU credits, there'll be a QR code available at the end of the webinar, and you can scan that with your phone and get your credit. If you don't have the capabilities to do that, you can either email Susan Brulette or type your name and CCA number into the chat, and she'll make sure that you get credits for that. I want to quickly thank all of our partners that have been involved in making this webinar series happen. I mentioned this on the first day. We've been working really closely with the conservation districts and other staff and members of UVM Extension and of UVM, and now of the new Soil Health Research Extension Center, the SHREC lab on campus. So very excited about that. And based on all of that collaborative work we've been doing, one of the things that we identified needing more attention were building knowledge around the indicators that we measure to tell us how healthy the soil is. So today we're going to continue in that series, and we're joined today by Dr. Deb Neher. And I don't know if everybody knows, but Deb recently retired, semi-retired, from what was the Plant and Soil Science Department at UVM. And when Deb started here, I was relatively new then, and she joined us as the chair of the department and then served as chair for many, many years. I'm not sure how long, Deb, but quite a while.
14 years. 14, 1-4.
Wow. And she was a great chair. She did a lot of great things for the department, and she's always been really supportive and also really involved and engaged with the Vermont community and also with UVM Extension. And I, for one, have always appreciated that. Deb is I think you're a nematologist, aren't you? Is that your official?
Well, I worked with nematodes. I call myself a soil ecologist, which, yeah. So it's I've worked primarily with nematodes and microarthropods, but I'm broader than that too.
Yeah. And she has taught a lot of students, graduate students, farmers, and lots of others as well. And we're really excited to have her joining us today to talk about something she's really passionate about, which is biological indicators of soil health, and hopefully helping us understand all of those options a little bit better. And I will turn it over to you, Deb. Thank you for joining us today.
Yeah, you're welcome. Make sure we got everything so you can see this.
Yep, you are up. Everything
awesome. Okay, well, I think you've seen this slide already in this series, but I put it in just as a quick reminder. When we got started, developing, our soil health programs on campus, we had a across campus, what we call a white paper exercise. And during that exercise, we had a survey of farmers, and two messages came through loud and clear. One is that farmers want tests that are better reflecting or more sensitive to management practices than they have been receiving. And the second is to add more biological tests. So that's been kind of our mission, or one of our missions. And so today I want to focus on those biological tests, and ones that we believe are some of the better ones to reflect management practices. Let's see if I can get my advancing to go. Okay. So I like to start out to remind everyone that soil is a living entity. Soil is alive. And it's not that we have organisms just living in soil, but they're actually a part or component of the soil. And, they can alter the appearance due to their biochemical and physical processes. And they also are very important in the functioning of soils. So just as a refresher on yesterday about the physical structure and aggregates, that biology is a very integral part of developing those aggregates. On the right-hand side, you can actually see a slice of soil. And this was from work we did in my lab. The blue is just a dye we put in there and represents the open spaces, the voids. But the black material there is in the lower left is a plant root. And you can also see organic material there. And the lighter color are just the mineral particles of soil. So you can see they're very integrated there physically. And there's a lot of spatial heterogeneity there. You've probably seen or heard of this kind of quote before, in terms of, you get this teaspoon of soil, and that there's more organisms in this, or actually 1 gram of soil, which is just a fraction of that teaspoon, has more organisms in there than there are people on Earth. The idea is these organisms are incredibly abundant and incredibly diverse. But today we have to be a little careful and not just stop at bacteria. And, there's a lot of different kinds of organisms that live here. So as you can see, we've got bacteria at the bottom, and that's what was being referred to in that teaspoon of soil diagram. Unfortunately, fungi aren't in this picture, but the soil also contains a lot of different animals or fauna. Some are microscopic in size, like protozoa, nematodes, mites, springtails, rotifers, etc. But then you can, work your way up. So I bring this because a lot of times when I talk with folks out in the field, I hear, well, I'm just measuring carbon, or I just need more microbes. But my point is those are pretty generic, and it's pretty broad brush, pretty general. It's a little bit more complex than that. And I'm hoping to kind of introduce you to some of those issues to help you appreciate that diversity and why we want to make sure not to lump everything. Now, one thing that's challenging about soil is that we haven't really had let me just back up to here just as a background, but soil biology is really considered kind of a last biotic frontier. We know more about the life on, in outer space and in the deep sea than we do in our own backyards. Only about 10% of the species in soil have been described. So, and why is that? One is we just haven't had tools to be able to look at these organisms in soil. And it's really been in these last couple of decades, well, maybe 30 years, with the advent of molecular tools and kind of nano-sized instruments that are even making it possible for us to gain insights as to who they are and what they're doing. So, as I said, first of all, microbes, some people just say that, and when I hear that, I just think it means bacteria. So we have to be careful. There's also a lot of fungi. But as I mentioned, there's other organisms that in that pyramid. I forgot to mention in that pyramid, the bigger the organisms, the fewer they are. It's kind of an inverse relationship. But I like to think of the soil community performing like a symphony. So, as we know with the symphony, not every player plays all the time. They come in at their prescribed time to do their task that fits in the music. So in reality, the organisms in soil, most of the species that are there are inactive most of the time. They're there, they're ready, they're all ready to act when needed, but they're in a dormant phase. So it takes the conductor, or we'll say that for soils, the conductor is the environment, and there's cues or signals in that environment that'll help wake up different groups at the different prescribed times. So, and that can depend on, is it dry or wet or warm or cold, or, a flush of nutrients or whatever, that can be cues. And then we have multiple species that play the same function or role, but they do them at different times or in different places or in different kinds of ecosystems. So I'm calling those the equivalent of the instruments. But I like this analogy because it's kind of showing how we need them to all work well together, but it's organized. Another thing is, a common metric of soil biology is just to look at respiration. And that's a pretty general term. It'd be kind of equivalent if I asked all these musicians to total up how much energy did they expend while they played their instruments, and then total that up and tell you. It doesn't tell me a lot about the music that they just played. It tells me a little bit about their effort. It's a first step, but there's a lot more to it. Another point is that we want complex food webs, and this slide is to remind us about how more complex food webs result in greater plant productivity. So what do I mean by a complex food web? This is a study. It was actually dealing with a grass, but it applies to other systems. A simplest system would just have a plant with sterile soil, or we could add bacteria, we could add predators of bacteria, we could put fungi in there, or predators of fungi, or we could put everything in there. And if you look over on the columns on the right, you'll see the number of the above ground biomass is the biggest one for that bottom row. And that's indicating the plant size. So we're seeing plant size, yield, plant productivity benefits from having a complex food web. So here's another way to kind of visualize that. I'm kind of adapting this from another study, but I think it's useful in this context. As we look at this diagram, we can see simpler food web on the left, and as we go to the right, it gets more complex. Now, agricultural systems by design, we have a lot of disturbances. We're putting plants in, we're adding things, we're cultivating, etc. And all of those are disturbances in the food system or in the soil food web, and tend to simplify it, those disturbances, as on the left. But what we aspire to is trying to get a food web that's more towards the right, and achieving that by reducing the amount of disturbances, thinking about, can we reduce some of that tillage? Can we integrate perennial plants into the system, etc. So our aspiration is for that complex food web, like on the right. Okay, Heather brought up on Wednesday about sampling. So I thought I'd throw in this slide just to fill you in, because she was exactly right in terms of sampling can vary depending on what it is you're measuring. And, my whole career has been working with soil food webs and biology. So I'll just fill you in on what we do. First, I'll draw your attention to the right-hand side, which is a nice illustration of the different scales of heterogeneity. There's 3 layers here. On the lower level is kind of large scale. So if you were looking at a field and the variations of soil type on a field scale, you can see the different colors represent different properties. And if we focus in on one of those that looks fairly homogeneous at the field scale and zoom in, we can see there's still a lot of heterogeneity within that. And then if we zoom in even more, we even see more like fine scale heterogeneity. So this is key, knowing that this heterogeneity is here and that these populations or communities tend to be clustered, they're aggregated and very heterogeneous, gives us a clue as to how we want to sample them. So if we're going out into a field and collecting cores, it's really important. The most recommended sampling patterns are, one is either for sure systematic. So it means a systematic would mean it might be, I have a random starting point and I take a core, I walk 15 steps and take another one, walk another 15 steps and take another one. The idea is you're doing it very systematic and regular, and trying to reduce the amount of bias. Another approach, and I think Heather had this one illustrated in her slide, was what's called kind of a stratified random, where you could divide up your field or site into smaller plots or areas and then pick a random point within that. The whole idea is that you're trying to make sure you represented all of this area without bias, and you want to make sure you're collecting cores in areas where it's aggregated, where there's a lot of organisms and equal chance of going between those clumps, so that you can get an overall estimate for the field. Just some pictures of us sampling this past summer. On the left, we typically use an oak field soil probe, which is about 1 inch in diameter. And for most things, we measure pretty much 8 inches, about 15 to 20 centimeters depth. The sampling depth really relates to what we're trying to target. If we're interested in plants we want to think about where are the roots, where are they most abundant. And so this depth has been adopted mostly because that's the depth of cultivation. But if one was more interested in decomposition, they might want to just sample the litter layer on the surface. Microbes, when you have these samples, you don't want them to get too hot or too cold. So it's important to get them into an insulated container right away, keep them in the shade. And if you're looking for bacteria and fungi, you would have an ice pack in there. If I'm looking at soil invertebrates, I do not want ice in there because they're very sensitive to it. So the idea is trying to keep them at a temperature that's similar to what you just pulled them out of in the soil. Oh, also with biology, you want to make sure all your instruments are clean between samples. You don't want clumps of soil left from the last one, because then that might be a biological contaminant into the next sample. So it's important to clean those out. So one thing I'll point out is looking at soil biology is laborious. And when it involves a lot of labor, it adds up in the cost. So we have to think very carefully about, what we want to look at and what we can afford to do. I just wanted to show you a variety of extraction methods that we've used in my lab. And, sometimes, first of all, it's very rare that you could actually collect soil and get a direct measurement from that soil. Usually you've got to do an extraction method to get it out of the soil first. The only case I see some direct measurements are if you can do some of the DNA samples on a small fraction of soil. So that's what the little centrifuge here is on the left, involves lots of pipetting. Microbes are so abundant in soil. Bacteria are on the billions per gram. 1 gram is about a size of a sweet pea. So we have to do a series of dilutions until we can get it dilute enough so that when we put it on a plate, we can actually see distinct colonies. If we have too many there, it's just a big blur and we can't get any counts. So this is an example of culturing. The culturing is a traditional method, but we've learned that you can only culture about 10% of the taxa in soil. So that's where the molecular tools come in, is we have to use other tools to get at the ones that we're not able to culture. If we're looking at soil fauna, like microscopic nematodes or microarthropods, we use methods like making a slurry, running it through a series of sieves. So that we do sieves, so they'll go through there, we can concentrate them, and then we can set them on some water and they'll swim out. Or for microarthropods, we use these heat lamps, and the samples are at the tops of these funnels, and they don't like heat and dry, so they run away, fall down the funnel into a container at the bottom. Sometimes we use bioassays, and that's what the plant is, especially for looking at plant pathogens or diseases. The plant is the gold standard, if it grows or not. Certainly, there's a lot of microscopy involved. It ends up that we have a lot of molecular tools for the pathogens in soil, but we're still in the infant stage of developing those for the free living or the beneficial organisms. So we still have to rely on a lot of microscopy. So techniques for identification and quantification can range from genetics and biochemistry. Those are especially needed for the microbes. And then morphology for some of the larger soil microfauna. And then, or it could be just measures of gases like respiration or estimating biomass. So I wanted to just introduce you to some of the players in the soil, in that food web, just to kind of show you the variety of entities. And these are all very common soil organisms. Before so the bacteria I've on the left-hand side and fungi on the right. Let's just start with bacteria. There, tens of thousands of species of bacteria in soil. That's my guess. We still don't know, but these are just a few general groups. We have Pseudomonas is a very common genus. There are dozens of species of Pseudomonas, and they have a variety of lifestyles. There's the Proteobacteria, another phylum, and one you've probably heard of is Rhizobium. These are the bacteria that live in the nodules of beans or other legumes. And those are little factories for nitrogen fixation. There's a phylum called Firmicutes, and this is where we have Bacillus strains. Bacillus subtilis is a common biological control. We know Bacillus thuringiensis, it's BT, it is used for controlling insects. And at the bottom, we have another group of bacteria called the actinobacteria. They used to be classified as fungi because they have hyphal-like structures. But in the meantime, we've learned they're actually bacteria. Based on the genetic material, the cell walls, etc. On the fungi, we have, I guess I didn't get a picture here of these two main groups. We know what yeasts are. They're just basically a non-reproductive fungus. A common one we know would be like Saccharomyces used in fermentation. There's the Chytridomycota, tend to be water molds, but then we get the groups called Zygomycota. These tend to be the kind you see on bread molds and decomposers. Rhizopus there. Alternaria tends to be a pathogen. It's in another group. Penicillium, Trichoderma. Penicillium I put in here to remind us that most of the antibiotics that we use in medicine actually originated from soil. And these organisms didn't produce it to control disease, but more as their own personal defense against other microbes in the soil. Trichoderma is a common biological control organism, and you can see it in culture or up close under the microscope. And then we have the mushrooms. But most of them in the soil are going to be the groups above that. And just for fun, I threw in this group of protozoa just to show you how diverse, how much there's functional diversity within the protozoa. Sorry. We have those, that are phagotrophy would be related to, with plants, or they can be in symbiosis, related to photosynthesis, or here as decomposers. One of the reasons I threw this in here was there's this group down here called the oomycetes, and this is a group of really important soil-borne pathogens that includes Pythium and Phytophthora species. which used to be classified as fungi, but now are actually classified as protists because they have these motile spores that move. And it's also because of the material in their cell walls. Okay, so then plants are super important to have in the soil. They are the base of the food web. And if you have plants there, that's the big reason, that's going to keep you keep them there consistently, and a variety of plants, that's going to contribute to a more complex food web. There's interactions, chemical communication between the plants and the microbes. This is a little drawing that one of the students in my lab put together to illustrate that. Plants give off exudates, which contain sugars and amino acids. They do this mostly because that helps their roots grow and lubricate their roots to enter into the soil, but those also serve as food for microbes. And this is, around the root is that really hot spot or concentration of these organisms. But you can get different variations. You can get some that are called commensal that really don't have any direct effect on plants, and they're not pathogens. You get some of them that we would call beneficial. They're the ones involved in nutrient cycling and uptake, or they might provide defenses against pathogens. And then there's groups of microbes in the soil that are pathogens themselves and can actually damage plants. Okay, that's the kind of background. I just wanted to give you a little microbiology 101 so we know kind of what we're talking about, have a visual, and have an idea of the kind of variety it's there. Now, I want to spend the rest of my time thinking about indicators, which was the whole purpose of this webinar series. Now, when we look at biological indicators in soil, I put them into about 3 different classes. Kind of activity, if we're just trying to look at general activity, if we're interested in who is there, and then what are they doing, which would be function. So under activity, this is where it would fit if we're doing like respiration would fit under this one. If we're trying to estimate biomass that's in the soil, enzymes. I mention enzymes because microbes cannot digest food in their bodies. They have to put enzymes outside of their body, dissolve their food, and then they can absorb it. So that gets us a little bit at that. But active carbon, and I put that in blue and as an asterisk there because that's one that we're planning to measure in Shrek. Then we have organisms, different ways. The one that I'll talk more about is what's a ratio of fungal to bacterial, fungi to bacteria. But that's getting us trying to separate a couple groups. One can do DNA, or we can look at microarthropods. It'd be other ways to get at that. When we're talking about function, we're talking about cycles of nutrients. And we could split that up into organic or inorganic forms, or we talk about the process of decomposition or plant productivity. So the ones in blue and asterisks are the 3 I'll talk more about that we're going to, initially measure kind of in our first rollout of Shrek. So it's 1 per category here. Before I go too much further, I want to mention, it's back to the laborious nature. Labor turns into cost. So ideally, it'd be great to measure all of these, but there's no way we can really afford to measure everything. So we need to pick which ones make most sense for our goals or what we're after. And what I tend to think about is try to think about how we can pick 2 or 3 out of these groups, but make sure they're complementary to each other and telling us different things. We don't want to measure 3 things that tell us the same thing, but we're trying to kind of like do the pyramid or the 3-legged stool. So collectively, they tell a good story. But it takes more than one to tell us the story. This was just a reminder that if we look in the literature, the most common metric that's been used historically has been looking at soil respiration or mineralizable carbon. These are number of citations. The two that I'm going to talk about a little bit more, ACE protein and POC, are gaining popularity, and partly because they're really responsive to management and they're cost effective. You can do them in a fairly time efficient manner, which keeps their cost down. And they're telling us more than what traditional measures of inorganic nutrients or for the soil chemistry has been telling us. This slide kind of shows that, well, actually, that's to remind me, one of the really traditional ways that we've done this in the agriculture and environmental testing lab, is to give an estimate of organic carbon. And we've done that based on a method called loss on ignition. We basically just put it in a very high temperature that burns off the carbon, and then we can subtract and figure out what the percentage of that weight loss is and estimate. That gives us a number, but really, if we want to get serious about, thinking about organic carbon, there's different pools of that carbon. Some are living, some are dead and decaying, and so forth. I'm not going to get into those details. I know the Monday seminar will get into more of the pools of carbon. I'm going to just kind of put it in to think about two kind of categories, kind of general categories. We have kind of an active pool or labile pool, meaning it would decompose fairly easily and readily. And then we have the protected pool. So when we're thinking about activity, what are those microbes doing? And in that sense, trying to reflect that, we're interested in that pool. But if we're concerned about climate change and carbon sequestration, we want to get at the protected pool. And another way is you can kind of think there's intermediate category in there between this kind of active and protective pool. And it's that intermediate category that we're able to get at with the method we're using called POC. So what is POC? It stands for permanganate oxidizable carbon. Okay. Permanganate and oxidation just happen to, reflect the measurement on how it's done. So, so we're getting an estimate of kind of the active organic matter, kind of this intermediate. So it's, you don't want that most active, because that's just going to burn off and you're going to get a lot of carbon dioxide due to respiration. You want something that's going to do it moderately so that you can still keep organic matter in your soil. You don't want to lose it too fast. You want to keep it there, but still maintain some activity. This measure is fairly simple. The big reason for choosing it is we found that it's much better suited to track management practices than the traditional measures of just simple percent organic matter. The bottom, you can kind of see a picture of the test. The purple is related to the permanganate solution, and the more of this material that's in the solution, the lighter it becomes. It represents, so it's kind of, change. It's measuring the oxidation state. So you have higher amounts of POC, the lighter the color. And we're able to quantify this using a spectrophotometer, which can quantify the color intensity. On the left, you can see there's a positive relationship of this method with some of the other techniques for looking at carbon. We're in the process of trying to develop, interpretation charts and ranges. And this is just a preliminary to kind of show where our range here is in Vermont. We have, score values and amounts here on the left, and we've got coarse. I believe these are different textures of the, coarse, medium, and fine soil textures, which impact the organic matter, as Joshua Faulkner was telling us about. And here's the entire range that's reported at a national level, where we're finding in the samples that we've collected in Vermont so far, we're just getting, just kind of in the initial phases of that, we're seeing, here's the range outlined here in green, and the purple represents kind of the mean of that range. Typically, the higher the number, the better. Just there are other people looking at interpretation. This is some data from a Michigan State long-term ecological research site. I won't get into all the versions here, but they have different cropping histories, cover crops or not, annuals and perennials. And we can see, if we're measuring mineralizable carbon, there's a big difference here in, this is the material that'll decompose pretty quickly and be lost as respiration. It's these values of mineralizable carbon are higher in these perennial systems than annual systems. And then here we can see that there's higher POC, a little bit higher here in the perennial than the annual systems. And then there's the differences in here have to do with whether or not they have a cover crop and which crop. So the point I'm trying to make is they are responsive to management. I'll skip that one just for the sake of time. We'll move on to, okay, so POC, okay, we're getting at carbon. The other measure we're doing is ACE protein. This is getting at nitrogen, one of our big nutrients necessary for plant productivity. Traditionally, in our, we've received information about inorganic nitrogen to give us fertilizer recommendations. And those forms are nitrate and ammonium, which are the forms plants can take up. But what that doesn't tell us is anything about the organic nitrogen. And so if, this has been kind of a missing piece of the nitrogen budget, as we haven't always quantified it. So now we're going to offer a test that's going to be an indicator of the organic nitrogen. So this is, what does that mean? Are these proteins? But it'd be basically you have decaying plant animal material, detritus. There can be protein in there. Proteins get broken down into amino acids. Amino acids then get converted to ammonium. So it's a pretty simple measurement, fairly time and cost effective, and has also been shown to respond to management practices. Here's just an example of it shown in that same Michigan State research site where they have, these colors are representing different annual systems. Then we have monoculture perennials in the orange, and then on the right, polyculture perennials. And we can see that this organic nitrogen is typically greater in the perennial, especially the polycultures where you have multiple species, a little bit more than the monoculture, one species in perennial, and both of those are greater than the annual crop systems here. We also, part of that is crops, but also another piece of that is tillage, that we see higher organic nitrogen ACE protein in the lesser tilled soils than the more tilled soils. And here's kind of the range of values that we found so far in Vermont. We don't find the full range that you see over the nation. It's kind of here in the middle, kind of shows the range and that. So we're still working on the interpretation of these as far as exactly, what we would expect. Like, here's the mark, the goal that we're aiming for. This is high or low for the state, but the first step is here to really kind of get the range that we would expect. And we're going to fine tune this. We have some research going on right now where we're actually have very detailed tracks of what the management practices are for each of these samples. And we'll then be able to start breaking out these values by management practice, which will be a lot more informative than just a number. So what are management practices we can do to increase the soil organic nitrogen? The main practices that are over here on the right, the things that we can do are to decrease soil disturbance, less tillage, trying to control erosion, trying to build organic matter, increasing crop diversity, and including cover crops in our systems. These are techniques that will help increase it. There are other variables we have less control over, and that, like soil texture or climate, but there are also factors there. The third technique that we're going to be rolling out is called PLFA. It stands for phospholipid fatty acid. So this is based on the idea that all organisms have cell membranes, and here's a picture of a cell membrane, and those membranes tend to have these they call it bilayer, meaning two layers. You can kind of see their kind of mirror images in a way, and you've got there's a the phosphorus is the head or the red, and then you've got these tails, which are fatty acids. You can see there's variety in these tails, and it tells us a little bit about, you know, the types of bonds between the different carbons in those chains. It ends up that different groups of bacteria and fungi and other organisms have unique signatures for these. So they'll have different fingerprints. So we can go about isolating these and quantifying it so we can get an idea of relative numbers of, say, bacteria and fungi. And so we're using this as an estimate to get an estimate of a ratio of fungi to bacteria, and generally aiming for more fungi representing a healthier soil. And why is that? Over on the right, I mentioned, you know, it's all about like what I would call succession. It's kind of a measure of disturbance. When we cultivate a field or add a large load of fertilizer or manure or something into the field, this is going to create a very simple food web that's dominated by bacteria. But if this is allowed to remain somewhat undisturbed and go through time, we start to see then fungi can eventually colonize, and they come in a little bit later in succession, and basically with a little bit less disturbance. So if we have higher fungal to bacterial ratios, it's telling us we have less disturbance, a more complex food web. This ratio correlates positively to some of the other measures that have been mentioned yesterday and today. It's pretty noisy, indicating, you know, in terms of these graphs, but that also is saying it's telling us something a little bit different than if you just went with the measures of available water or aggregate stability or soil organic carbon. Here's some data just to show how tillage affects this ratio. We have the proportion of biomass composed of fungi on the y-axis. We have soil water content on the x-axis. The line with the solid symbols is no tillage, and the open symbols are conventional tillage. We can see the values with no till have higher ratios of fungi to bacteria than do the cultivated. You can also note that it also has more water holding capacity as well, which is further to the right on this graph. Just to give you an idea of what we're aiming for, you know, interpretation is something a bit like this. The Ward Lab offers a ratio. But we want to, you know, provide a scale that's specific to Vermont. So where these cutoffs for these different levels will be changing as we build on our database for the state. Now I just wanted to just briefly mention a few aspirational indicators that would be nice to do in the long term that I think would be very useful. We can only do so much so fast. So these are just aspirational at this point. I'd mentioned enzymes are important. These are proteins that microbes send outside their body to digest their food. The value of having enzymes is they're telling us a little bit more about function or what these microbes are doing. And here's a picture of the nitrogen cycle on the right-hand side. And you can see there's different steps in the nitrogen cycle that have different conversions. Each one of those steps is catalyzed by a different enzyme, and different microbes produce different suites of enzymes. But if we know enzymes, we can kind of get an idea of where in this process or what processes are happening. Or we could get an idea, are they decomposing lignin or cellulose, or is it urea? You know, if we can tell this based on enzymes. Due to time, I'm just gonna, well, I'll just briefly do this. This is put out by the Soil Health Institute. And this is a metadata analysis where they've looked and compiled a lot of studies together. On the y-axis are different management practices, whether it's a rotation, crop numbers of crops, residue retention, organic amendments, cover crops, or decreased tillage. And then there are 6 different measurements here. And if they're on the left of this line, you see there's a negative relationship. If the values are on the right-hand side, it means there's a positive relationship. So let's say respiration here, positive, you know, more respiration with residue retention, organic amendments, cover crops, and less tillage. But you can see similar patterns over here if we looked at the enzymes that break down cellulose, the enzymes that break down chitin, and so forth. But we can see there's definitely different impacts by the different management practices. I'm going to put a plug in for soil fauna just briefly. I think this is an aspiration. It tells us a little something different than the microbes. What are the reasons? They're higher in the food chain. I've highlighted the fauna here. We've been talking solely today about the primary decomposers, which is bacteria and fungi, but this group are the ones that feed on or are predators of those bacteria and fungi. So it gets us more at a predator-prey thing, higher in there. The generation time of these organisms is a little bit longer. It's more like days, or I mean, longer like days to years, where microbes can reproduce within hours and days. So it's a little bit more stable temporally and not just an ephemeral response. And through some research I've done, noted that soil properties and microbial biomass really only predict about 23% of the variation associated with the invertebrate communities, these soil fauna, microscopic soil fauna. So they're indeed telling us something very different or complementary to microbial measures. Just to illustrate what I mean by different is this little table on the lower right-hand side. Very simple experiment. These happen to be with bacteria, bacterial feeding nematodes, and then mites, Tideid mites. And we're looking at decomposition. We have a control system or adding insecticide. So what the goal is, trying to look at measuring decomposition. And here we can see, you know, we have 10 to the 7th bacteria. We've got this many 6.6 of the nematode, bacterial feeding nematode, and then we have mites. Okay. We add insecticide, so we knock out all the mites. There's no more. We remove a trophic level. We see now an increase in the bacterial feeding nematodes because they just lost their predator. And if we have more of those, there's fewer bacteria by an order of magnitude of 10. And if there's fewer bacteria, we have less decomposition, less of the function. So when we just look at bacteria alone, we have to keep in mind, you know, there may be something above them that's moderating those. If we get low values, it might just mean that there's a lot of predators feeding on them. When we look at nematode communities, there's, you know, multiple trophic groups that are in the center of the food web. The concept that I was describing of bacteria to fungi as kind of an ecological succession event for that ratio also applies to the nematodes. I just illustrate what we know about plant communities where you have highly disturbed primary succession to later. There's indices that can reflect this in terms of certain nematodes that come in and are very tolerant of disturbance, others that are very sensitive to disturbance. And we can look at the community and compute kind of the relative amount of each of these to say something about how much disturbance and what kind of disturbance. I'm gonna skip a couple slides because I want to make sure we have time for questions. Kind of my last point for an aspirational is when we're looking at PLFAs, we're just getting relative amounts, okay? And it's kind of estimated on biomass. But if we really want to get at who is there, understanding the species and taxa, we have to get down to sequencing. And so we can sequence, you know, I won't go into the details of it, but there's certain gene regions that we're able to target that have a lot of diversity among the different types of bacteria and fungi. It can be very powerful to tell us about who is there. And so a profile for soil might look something like this. If who is there, they can tell us, here's our different phyla. It's a very coarse level of different bacterial groups. And important, you can just look at the color differences. These are just two different management practices where they had a legume in the system versus adding synthetic fertilizers with nitrogen, and how there's some adjustments happening with how they are affecting this bacterial community. Okay, that tells us who's there, but what it doesn't tell is what they're doing. And that's kind of the state-of-the-art research in soil biology, is trying to learn what they're doing. And the way that we achieve this is we use genes that we know are related to specific chemical reactions. And then they can look at particular genes and determine what is happening, say, in the cycling of nitrogen. All these little abbreviations around here represent different genes that can be picked up, and they can inform us as to, like, let's just say you had a lot of this NIRK up here. If the taxa had that, then we know that one, what it's doing is converting, you know, the form here of nitrogen to this other one on the left. So it can tell us exactly what its role is in the nitrogen cycle. So just to recap, food webs are complex. Complex food webs are healthier than simple food webs. And it's more cost effective to choose complementary indicators, get a small suite of those, than doing large numbers that are repetitive. We have to keep in mind the interpretation of microbes depends on what's happening above it as a trophic group. And the initial offerings we'll offer in the Shrek lab are POC, ACE protein, and the PLFA for the fungal to bacterial ratios. Now I'm going to stop there and let you ask some questions, because I'm sure I have some holes in this.
Great, thanks, Deb. That was awesome. Very interesting.
Good.
I found the slide that you showed with the different trophic levels. I just want to make sure I understood what it was showing. You had the top trophic level of mites. I think it was mites.
Okay, let me see which picture you want.
Eating nematode. Or
was it early on?
Nope. Go back a little bit. Right there. Oh.
Oh, that one.
It showed
with that little yellow table right there.
Insecticide. Yeah.
Yeah.
So I've seen this in some of our data.
Mm-hmm.
Oh, mites. Is that what I said? Yeah, mites.
Yep.
So this is showing quicker organic matter loss?
With mites, yes.
With mites or without mites?
With mites.
8.2. Okay, so
you can see here, when you have mites present, you had, say, 23% organic matter loss. When they're absent, I was about a third of that.
Okay.
Yeah.
Yeah.
So the idea, I guess I didn't explain very well, is by having multiple trophic groups, having this trophic complexity like that, there's actually more regulation happening that kind of controls this decomposition rate. And by having predators and secondary predators, yes, the role of predators is they call out the kind of the sick and the old, or so to speak, and keep the community really vibrant and alive and active.
Yeah, no, it does make sense. I guess part of what I was thinking is that the organic matter loss is slower, but there's probably also less organic matter in the insecticide system, or we don't know.
Oh, well, that's a good question. This data doesn't tell us how much they start. Well, this is just telling us the rate of decomposition.
okay.
It didn't tell us how much, but you're correct. Likely in that long-term system, you would likely, well, it depends.
Yeah, I
I
guess I was
guess,
just thinking
yeah.
about maintaining a biological system that's really active and complex. Do you need to have more organic matter additions to keep up? You know, it's sort of that
It's kind of a
if
chicken and egg
you
thing.
right, if you build it, they will come, but once they come, do you have to keep adding, because they're cycling organic matter faster, which means nutrients are cycling faster.
And you're losing your organic matter faster.
Yeah, anyway, I think I was thinking about it
Yeah.
too deeply. Anyway, we'll get
Well, it's interesting with these guys because another question is where do they come from?
yeah,
Right. So,
right.
Adding organic matter is providing their food. There can be some there, but they might be hanging out on the margins of your field, and on other plants, your weeds and so forth. And they actually, when there's food there in your field, they'll come. They
Yeah.
migrate in there. So it's kind of like, what do they do when there's no food left in the, the organic matter is all gone. But it's amazing how if you keep other plants around it and your margins, this is where, keeping those margin communities, they're actually kind of your seed bank, for these organisms to come back in.
Paul had a question around the underwear test, which I'm sure you've seen where people bury
sure.
the cotton underwear. And he's wondering if this simplifies all this. He Paul, you call them the Paul factors.
Well, that's you're getting that decomposition rate. So that's getting at that. It's not telling you who's doing it. You don't have any idea of the community composition, but you are getting at function. And one of the values of that test, and there's also teabag tests and different things, is they're trying to figure out, if you want to get citizen scientists or think about global, you're trying to get something that everybody has. So you can have a common substrate, and then I can compare my data with your data and Paul's data.
Right. That's good.
And underwear is good because it's cotton usually. And you want 100% cotton so that you know what you're decomposing. If you had polyester blends, that would be a different thing because those don't break down so well.
Right. Another question that came in is about if results from our soil at different times of the year varies. So no sampling in the winter when frozen, or is there a whole other group of life going on then?
Okay, now repeat that question.
Yeah, I think the question is, if we're always sampling at the same time, which has kind of been the recommendation.
Oh, yeah.
Are we missing a group of organisms? Like, let's say we did go out and sample in the winter. Would we see something?
No, you're getting different things. So that's the symphony idea. You're going to get some you might get there are definitely seasonal effects, and there's definitely plant phenology effects. So when the plant is a little seedling, it's going to be different than when the plant is sending out lots of roots versus when it's putting out its seed in the yield or at the end. So, I mean, in the ideal world, you want to sample at different times. But if you only can afford, and this is often the case, scientists, we should be sampling multiple times. But if you're the user, it's tended, and this is slanted by people looking at pathogens. People tend to do it near harvest because that's often when the plants are the most susceptible. It's had the whole season. You can see root damage, and they're usually at their peak numbers. That would be if you're looking at parasitic organisms. If I am recommending it, where I've narrowed it down to 3 times, for looking at season, and that's usually at planting, at the mid-season, like right in that transition between vegetative and reproductive, because that's when the roots stop growing, and then at the end of the season.
Great, thank you. And I see Sean has added in some comments too about seasonal variation with POC and yeah, just really thinking about when you're sampling. And I also,
and we're
go
seeing
ahead.
those seasonal differences in the samples that we collected this summer on these at these different farms. We can definitely see changes in the season. We did those 3 times that I mentioned.
Well, it is 1 o'clock. And I think, you know, we started off in the first day saying there's more that we need to know, which is why we wanted to have the webinar series, because we had a lot of conversations around these indicators after taking all these samples, trying to understand them and make sense of them. And when we started this webinar series, I assumed that we'd end up with more questions than answers by the end too. So it's definitely an emerging field and we have a lot to learn.
I often say for every question I answer, I generate 10 more questions.
Yeah. Now everybody knows there's a lot of questions to be answered.
So I'm just at the tip of the iceberg, and I hope it at least helps set a foundation for future webinars we have. Oh, good.
I really enjoyed it. And thank you so much, Deb. And thanks everyone for joining us today. And hopefully we'll see you on Monday for the wrap up. We do want your feedback on other education you're looking for on this topic. As we've mentioned, Shrek has an E in it for extension. So it's not just research and it's not just a lab. We want to try to connect all of these pieces together and provide the education and outreach that folks need on soil health. So if you have other topics you want to learn about, we want to hear about it. So please fill out the survey and or just email one of us, myself, Lindsay, Susan, Deb, Joshua. We all want to hear your thoughts on this. So thank you again, Deb, and thanks everyone.
You're welcome.
Have a wonderful weekend and we'll see you on Monday.
All right. Thank you for the
Thanks, Deb.
opportunity.
Yeah.